and it's a great pleasure for me to be here to talk to you today about understanding in vitro folliculogenesis in farm mammals. I'm going to cover the following topics. The first, an overview of folliculogenesis regulations in vivo. Then, some basics and applications of in vitro follicle culture and its use as a tool for understanding folliculogenesis. Focus on the balance between extra and intra-ovarian factors, influence of extracellular matrix, follicle interactions, stage-specific follicular requirements, and eventually some final considerations. It's well known that mammalian ovaries contain from thousands to millions of follicles, whereby approximately 90% of this population is represented by preantral follicles, which in my definition includes the primordial, intermediate, primary, and secondary follicles. Despite this large follicle population, the vast majority of them will be eliminated by a physiological process named atresia during folliculogenesis. Folliculogenesis is the physiological process in activation, growth, and maturation of the ovarian follicles. We define follicular activation being the transition from primordial to growing follicles. The regulation of folliculogenesis involves a complex interactions among endocrine, paracrine, and autocrine factors, which in turn affect steroidogenesis, angiogenesis, basement membrane turnover, follicular atresia, oocyte growth and maturation, as well as the proliferation and differentiation of follicular cells. Folliculogenesis is a such complex process because follicles in the ovaries are under influence of many extra and intra-ovarian factors. To make matters worse, many of these factors share the same receptors. Also, there are complex interactions among cell signaling pathways that eventually control gene expression, which determines cell survival or death, quiescence, or proliferation. Therefore, follicular fate in vivo, atresia, or ovulation depends on a fine balance between stimulatory and inhibitory factors. After this brief overview, it's reasonable to state that understanding the control of early folliculogenesis is crucial to optimize the use of ovarian oocyte reserve in assisted reproductive technologies in human, as well as in other mammalian species. Among these technologies, I will highlight in this presentation the in vitro follicle culture. Now, I'm going to give you some basics and application of these technologies. In fact, taking into account that the vast majority of follicles in the ovaries will be eliminated by atresia in case they remain in the ovary, the ultimate goals of in vitro follicle culture are rescuing preantral follicles from the ovary before they become atretic, being able to culture them up to maturation states for the purpose of preventing follicular atresia. The aim of this technique is to mimic what happens with a few follicles that escapes from atresia during the folliculogenesis and ovulate. Then we also name this technology artificial ovary. This technique has some future and current application, such as study the control of early folliculogenesis, complement other reproductive technologies, create gamete banks from endangered species and breeds, preserve the fertility in individuals subject to cancer treatment, human infertility treatment, and also aid in studies on reproductive toxicology. Goats, the many species I work with in Brazil, are considered a good model for human due to the similarities in terms of ovarian size and texture, as well as they share long-term folliculogenesis, approximately 6 months. In Brazil, we have successfully used goat in vitro follicle culture to perform toxicity tests of anti-cancer drugs for use human, such as doxorubicin. Basically, there are two ways to culture follicles in vitro, in the isolated form or enclosed in ovarian tissue. We name this in this presentation in situ culture. Isolated follicles can be cultured either in a two-dimensional system, which means the follicle is placed on the plastic, which may be a plastic or an extracellular matrix, such as collagen, or in a three-dimensional system enclosed in an extracellular matrix. In situ culture, on the other hand, can be performed using an ovarian slice or the whole ovary. The efficiency of in vitro follicle culture can be evaluated using the following parameters: follicular survival, follicular activation and progression through folliculogenesis, oocyte and follicular growth, hormone production, gene expression for key factors, ligands and receptors, entry formation, production of full-grown meiotically competent oocytes, and we can also evaluate oocyte developmental competence through the production of embryos and live offsprings. It's important to emphasize that the production of live offsprings from primordial follicles cultured in vitro has been only achieved in mice, and it was first reported in 1989. In order to develop an efficient culture system for caprine preantral follicles, since 2006, we have been working a project named Artificial Ovary. In this particular project, the effect of more than 30 factors in different concentration, association, type of culture system, culture addition time, and culture duration are being tested. In the next slides, I'm going to share with you a few protocols and results from this project. Now, I'm going to show you the use of in vitro follicle culture as a tool for understanding in vitro folliculogenesis using the GOAT model. Let's get started with the GOAT, primordial follicle survival and activation. To study this, we have been using the in vitro culture of caprine preantral follicles enclosed in ovarian tissue. Basically, ovarian tissues from slaughterhouse material have been cultured for 7 days at 39 degrees Celsius in 5% CO2 in air in a modified minimal essential medium. This table summarizes the main results of the test. As you can see here, the addition of all these factors individually to a control medium improved significantly in a concentration-dependent manner follicular viability, activation, and growth. Similar results have been obtained using preantral follicles from cow, pig, sheep, and mares as well. It's very similar, in fact. As you can clearly see here, many factors improve the same endpoint, which can be explained by the fact that all these factors will end up controlling gene expression, which determines follicular fate. Now, I'm going to show you an example of long-term in vitro culture to develop sequential media. In these protocols, ovarian fragments were cultured for 16 days in a control medium or control medium supplemented with 2 factors, A and or B, added in the first and or second half of culture, resulting in 10 treatments. Just to give you an example, we tested the combination of FSH and GH, or GDF9, or KL. The best outcomes were obtained using FSH in the first half, followed by GH. I'd like to draw your attention to the highest percentage of follicle survival, activation, secondary follicle formation, and follicular and oocyte diameter in this treatment. Now, I'd like to turn to the use of in vitro culture of secondary follicles to study the balance between extra and intra-ovarian factors. Secondary follicles were isolated mechanically from the ovary using a microdissection procedure and cultured individually in 100 microlipid drops of a medium without antibiotics. After the test, the best outcomes were obtained using GH, FSH, insulin, and VEGF. This slide illustrates an example of experimental design to develop sequential medium using bovine recombinant FSH. In this protocol, isolated follicles were cultured in a control medium or control medium supplemented with 2 fixed FSH concentration, 100 and 1,000 nanograms per ml, or in sequential medium 1, 3 increasing FSH concentration added in different culture times, or sequential medium 2 different FSH concentration. Compared to the control medium, the best outcome for follicle survival and development was obtained using sequential medium 1. When we used the control medium containing increase in FSH concentration, and we supplemented this medium with LH plus EGF or GH alone or VEGF, we succeeded in producing the first ever caprine embryos from isolated secondary follicles cultured in vitro. Okay, just to illustrate this achievement, in fact, this is a caprine secondary follicles at onset of culture. We stimulate these follicles with GH, and here you can see the anther formation, the formation of a mature oocyte. You can see the first polar body here, and an embryo at modular stage. In the previous experiments, the maturation rates of in vitro grown oocyte were similar between treated groups and the control group. This problem was overcome using an appropriate VEGF concentration. In fact, VEGF at 100 nanograms per ml increased significantly the percentage of full-grown oocyte, meiotic resumption, and metaphase 2 when compared to the control medium. Then, one question has arisen. Does the insulin concentration affect the in vitro culture of goat preantral follicles? In other words, was the insulin concentration currently used for the in vitro culture in many studies, I mean, insulin at 10 micrograms per ml, considered high, the best for goat preantral follicles? To answer this question, isolated follicles were cultured in a control medium or control medium supplemented with 3 insulin concentration, 5 and 10 nanograms per ml and 10 micrograms per ml in the absence or presence of FSH. Insulin at 10 micrograms per ml reduced significantly the percentage of follicle survival and formation, but increased follicular diameter when compared to the control group. This negative or positive effect were not observed when FSH was part of the culture medium. However, insulin at 10 nanograms per ml showed a higher percentage of meiotic resumption when compared to the control medium and high insulin. In other words, we reduced the insulin concentration in 1,000 times, and we improved the percentage of meiotic resumption. Insulin plus FSH also increased significantly the mRNA expression for insulin receptors, aromatase, and also estradiol production. Up to this point, we had 2 situations. A medium contained VEGF, increase in FSH concentration, and high insulin that provides the best outcomes for oocyte maturation, and another medium containing increase in FSH concentration associated with low insulin. The point was, could you just replace a high concentration of insulin with a low and maintain the efficiency of VEGF medium? The answer is no, because it's not that easy. When we compare two concentrations of insulin alone or associated with FSH, fixed or increasing FSH concentration, in a base medium containing VEGF, the best outcomes were obtained using high insulin associated with fixed FSH concentration. Similar results were obtained when GH was part of the base medium. Therefore, when VEGF, in fact, is part of the base medium, we need to reduce FSH concentration and increase insulin concentration. Now, I'm going to discuss with you some results related to the influence of extracellular matrix on in vitro culture of isolated caprine preantral follicles. This research has been done in collaboration with Woodruff's lab here in Chicago. In this protocol, in fact, preantral follicles were cultured in a two-dimensional system on plastic or in a three-dimensional system using 3 alginate concentration. Compared to the control group, higher alginate concentration were better to preserve follicular survival, while low alginate concentration provide the best outcomes for follicle growth rate and percentage of meiotic resumption. Then we used the alginate 3D system to study follicle interactions in vitro. In this protocol, we compared single follicle culture, which means one follicle culture in one alginate bead, with multiple follicle culture, where group of 5 follicles were cultured in individual alginate beads or in the same alginate beads. The results showed that multiple follicle culture in the same alginate beads showed a significantly higher percentage of antral formation compared to the multiple follicle culture in individual beads and larger follicular diameter compared with single follicle culture. Then, in the next experiment, groups of 5 follicles were cultured in a 2D system or in a 3D system in the presence of alginate, fibrin alginate, and hyaluronate. The data show that compared to the control 2D system, fibrin alginate provide the best outcome for full-grown oocyte percentage of meiotic resumption and metaphase 2 oocyte. This data clearly show that the type of extracellular matrix does affect the in vitro follicle culture. And when we culture these follicles for 30 days in fibrin alginate, we succeeded in producing a metaphase 2 rate similar to the in vivo grown oocytes. And we also produce a few embryos after parthenogenetic activation of metaphase 2 oocytes. Now, I'm going to show you some results related to stage-specific follicular requirements. I do believe that follicular requirements change as the follicle develops from primordial to pre-ovulatory follicles. To confirm this statement, in collaboration with Dr. Gastal of Southern Illinois University, we investigate gene expression in late secondary follicles and early tertiary follicles in goats using microarray analysis. Despite these 2 follicular categories differed only in 150 micrometer in diameter, we found that 2,400 genes were stage-specific up and down regulated in the transition from secondary to tertiary follicles. In vitro culture studies confirm these findings and show that preantral and antral follicles behaved differently to human FSH and insulin stimulation. In fact, addition of FSH stimulates follicle growth rate in antral follicles, but not preantral follicles. However, difference between low and high insulin was only observed in preantral follicles. Moreover, GH addition to a base medium containing low insulin increased oocyte diameter, meiotic resumption, and metaphase 2 rates only in antral follicles, but not preantral follicles. Then we use the GH medium to try to figure out a non-invasive follicular sign, the relationship between non-invasive follicular sign of caprine preantral follicles and meiotic competence. Then early antral follicles were cultured for 12 or 18 days, and then oocyte diameter, viability, and chromatin configuration were checked at day 0, after 12 and 18 days of culture, before and after individual oocyte maturation. In this protocol, it was very interesting because thanks to single follicle culture and individual oocyte maturation, we could match each oocyte during in vitro maturation with its corresponding follicles and study consequently the relation between follicular dynamics during in vitro culture and oocyte individual maturation. The data showed that, surprisingly, at day 0 of culture, 14% of the oocytes were already degenerated, and 9.5% resumed meiosis when released from the follicles. We also observed that oocyte diameter increased significantly from day 0 to day 12, but did not differ from day 12 to day 18. However, only oocyte from follicles cultured for 18 days increased significantly in diameter during in vitro maturation and provide the best outcomes for full-grown oocyte, percentage of full-grown oocyte, and metaphase 2 oocyte. And we also found positive correlations between oocyte diameter, follicular growth rate, and oocyte meiotic competency. And we also observed that follicles that reached 700 micrometer in diameter showed a higher percentage of metaphase 2 oocyte when compared to the other follicular size. Now, I'll leave you with some take-home message. First, balance between FSH and insulin concentration in a medium containing GH and VEGF is important for oocyte meiotic competency in goats. Second, follicular requirements differ between preantral and early antral follicles. Third, preantral follicles cultured in group in fibrin alginate showed metaphase 2 rates similar to in vivo grown oocyte. Fourth, it's possible to establish a non-invasive sign for efficiency of in vitro follicle culture based on follicle daily growth rate and diameter. And finally, the in vitro embryo production from goats grown oocytes grown in vitro is feasible but inefficient, probably due to the inadequate oocyte cytoplasmic maturation. I'd like to thank some partners and sponsors, especially CNPq Brazil, for the main financial support in this project. And I'm really deeply grateful to my research group in Brazil for their dedication, motivation, and for doing the very hard job in this project. And I'm very proud to say that in our lab, in addition to scientific education, in association with a nonprofit Christian organization, my team members are involved in service learning project to help vulnerable people located in the poor rural region in Brazil. Last but not least, I thank you all for your kind attention. Thank you very much.
Thanks, Ricardo, for the wonderful presentation. This is a lot of data. 30 minutes, it's just a little to show everything you have done at your lab. And Teresa, do you have a question?
Yes, what a brilliant presentation. Thank you so much. My question has to do with the complexity of the signals and the fact that many of them converge on common signaling pathways. Have you tried to use just agonists, so receptor-independent agonists of different doses at different times? So example, forskolin, so that you could eliminate the differential ligands in favor of perhaps different cocktails that would be changed with time that are simple agonists for the interior cascades that we're trying to activate. Have you thought of doing that?
It's a very good idea, a very good idea. But we are trying now to use the ultra-diluted drugs in culture, to very, very low concentration of drugs. It's a project that is going on.
I have one question. If you already did in vitro follicle culture using vitrified or cryopreserved follicles, and if yes, if the response like the fresh ones?
The results are very similar, in fact. Ana Paula, it's a colleague there, have been doing this kind of experiment. It's worked pretty good.
Okay, thank you.
Mary?
Ricardo, thank you very much for your presentation and all of the work that you can do with the animal model that you have that saves us a lot of work that we need to do. 2 questions. So in the non-human primate, we see follicles that don't grow, follicles that grow fast, follicles that grow slow. Do you see the same thing in your caprine model?
Yes, it's the same.
Okay.
Yes.
And have you ever measured AMH concentrations in the media?
No, not yet. But it's a possibility to do.
Okay, thank you.
Thank you.